Hi everybody, thank you for coming back. We'll start now the session number 3, which is experimental treatment. First lecture will be given by Sonia Jerez from Spain. Sonia is a scientist at the IVF Foundation in Spain and member of the Reproductive Medicine Group at the Institut Investigador Santiago Lefe in Valencia, Spain. The topic that she will discuss with us will be bone marrow-derived stem cell in women with diminished ovarian reserves and premature ovarian failure. The second lecture will be given by Elena Labarta, also from Spain. Elena is a physician at the Valencia Institute of Infertility, the IVI, in Valencia in Spain. She will present the topic Mitochondria as a tool for oocyte rejuvenation. The third lecture will be given by Evelyn Telfer from the United Kingdom. Evelyn is working at the Institute of Cell Biology and Center for Integrative Physiology at the University of Edinburgh in the United Kingdom. She will discuss with us Orgonia stem cell. The fourth lecture will be given by Kazuhiro Kawamura from Japan. He will present the topic in vitro activation of follicles for infertility treatment in poor ovarian reserve. He is the director and associate professor of Reproduction and Infertility Center of St. Mariana University School of Medicine in Japan. The fifth lecture will be given by Mina Quidberry from the United Kingdom. She is a consultant in gynecology and reproductive medicine at Newcastle Fertility Center at Life Science. Her presentation is about the preventing mitochondrial DNA disease, pronuclear transfer. Summary of the session will be given by Evelyn Telfer at the end of the session. So we will start now with the first lecture.
So first of all, I would to thank to the organizing committee for allowing me to present our work in this very innovative meeting. As all of you know, the depletion of the ovarian reserve is characterized by a progressive decline in both the follicular pool and the AMH levels. Nevertheless, there are some situations where this decline in the ovarian function could be accelerated or totally impaired, like in patients with diminished ovarian reserve or POI patients. Which are the therapeutic alternatives? So nowadays, most of them, especially for the poor responders, are focused on the stimulation protocols. But no matter which protocols do we use if there's no antral follicles to be stimulated. There is one thing that should be highlighted, and is that even in POI patients and menopausal patients, there are a remaining pool of dormant residual follicles in the ovary, even when the ovary loses the ability to ovulate or to function. So we felt that we should put our attention in this population. In fact, follicular rescue and live birth after in vitro activation and ovarian fragmentation in these POI patients was demonstrated by Kawamura and colleagues in 2013. So these residual follicles are able to grow and be ovulated in an adequate ovarian environment. In fact, we felt that this follicular rescue could be the underlying mechanism of the several reports of fertility recovered and live births in infertile patients due to chemoradiotherapy after the bone marrow transplant. The bone marrow-derived stem cells represents an heterogeneous group of mononuclear cells, which includes several progenitor or stem cells, hematopoietic and also mesenchymal stem cells. Can be produced and mobilized to peripheral blood for collection by a pharmacological treatment. We are going to have always an autologous source, and we don't need cell culture to assess enough cell concentration. So in order to evaluate our hypothesis, we first established a mouse model of diminished ovarian reserve and POI. And then, once the ovarian damage was established, the animals were were infused with the human bone marrow-derived stem cells. And we observed that the infusion of bone marrow-derived stem cells, when compared to normally circulating blood cells, was able to restore the estrous cycle and estradiol secretion, even in the POI model, promoting the follicular development and increasing the number of preovulatory follicles. We also observed an increase in the number of metaphase 2 ovulated oocytes and early embryos, and also a long-term restoration of the spontaneous fertility in the treated groups. If you can see in the graph, the animals in the POI control group were not able to reach any single pregnancy during 3 months. But by contrast, animals treated with the bone marrow-derived stem cells were able to reach several spontaneous pregnancies and having healthy pups. We also observed that all these positive effects on the follicular growth were mediated by the regeneration of the ovarian niche, where we observed an increase in ovarian local vascularization, cell proliferation, and a reduction in apoptosis. In a second step, we aim to validate these results by using human ovarian tissue. With that purpose, we xenographed ovarian cortex from poor responder women into an immunodeficient mouse model. And we observed that in human ovarian tissues from poor responder women, these bone marrow-derived stem cells, when compared to the CD133 positive cells, which were also tested in these experiments, as these are the population used by Dr. Simon group for Asherman patients, were able to engraft into the human ovarian tissue close to vessels and even in contact with granulosa cells. Also promoted the development of human follicles, as secondary follicles were only observed in those grafts treated with the stem cells, being the higher amount in the bone marrow-derived stem cells rather in the CD133 positive cells. In the case of human ovarian tissue, we also observed a regeneration of the ovarian stroma by increasing cell proliferation, especially in the granulosa cells, and enhancing the ovarian local vascularization. So, we felt that the human bone marrow-derived stem cells rescue the already existing follicles to growth and to produce healthy offspring. And this follicular rescue could be promoted by the regeneration of the ovarian niche, as suggested by the increased vascularization and proliferation. So maybe the bone marrow-derived stem cell-based therapy could be a suitable alternative to increase the reproductive potential of women and patients with diminished ovarian reserve or impaired ovarian function. Having all these results on mind, we designed an alternative treatment for women for follicular rescue of these remaining follicles by the autologous stem cell ovarian transplant, or the Ascot technique. In a first step, we developed a pilot study with 20 poor responder patients diagnosed following the ESRO criteria. The Ascot technique has 4 steps. The first one is the mobilization of the bone marrow-derived stem cells by a 5-day treatment with granulocyte colony stimulating factor, following the same protocol that is used in the hematology field for bone marrow, healthy bone donors. Then, in a second step, the bone marrow-derived stem cells were isolated from peripheral blood by apheresis, and the cells were infused by intraovarian catheterings into one ovarian artery. After the treatment, our patients developed a 6-month follow-up for the ovarian reserve biomarkers, AMH and antral follicular count, and when was possible, we also developed an in vitro fertilization study. Our results show a high variability among patients for the AMH response, but we identified 2 peaks of positive response for AMH. The first one was detected around days 15 or 20 after the treatment, and the second one appears like 3 to 4 months after. Regarding the antral follicular count, we observed that the total antral follicular count significantly increased on days 8 to 22 after the stem cell treatment, being especially significant 2 weeks after the cell administration. Positive response of ovarian reserve biomarkers was observed in 81% of the treated patients when considering a positive response like an increase in at least 3 follicles for the antral follicular count when compared to the baseline levels for each patient, which were established prior to treatment, or an increase in the AUGMENT serum levels in 2 consecutive determinations. But no differences was detected when the control and the treated ovaries were compared. We also observed that this positive response in antral follicular count, or AMH, was correlated with higher concentration on the apheresis of the stem cell secreted factors, the fibroblast growth factor 2 and thrombospondin. Then we start the IVF study. It's important to highlight that 2 patients were excluded, so only 18 of our recruited patients completed the whole study. For this part of the study, we use exactly the same stimulation protocol than the patients used in previous cycles, because we aim to have only the stem cell infusion as the only difference. We also performed the PGTA analysis in order to see if there is any positive effects on the euploidy rate of our embryos. When the previous cycles and the cycles developed after Ascot were compared, we observed a significant increase in the AMH levels, the antral follicular count, as well as in the punctured follicles. And we also observed a reduction in the cancellation rate, which is one of the main problems in the stimulation of poor responder patients. Then we consider some of the cycles as the optimum ones. Why? Because they were started 2 weeks after the stem cell treatment, when we observed the antral follicular count. And in this specific subset of cycles, these improvements were even higher, reaching a 0% of cancellation rate. The overall IVF outcomes show that 85% of the started cycles were able to reach the oocyte pickup, having metaphase 2 in 66% and embryos in 65%. If you see on the table, for having similar number of cycles, the number of metaphase 2 oocytes and embryos were almost double. But we observed one problem. Only 7 of the 40 embryos obtained were euploid. We don't have previous data for euploidy because these patients do not perform PGTA in previous cycles. But this increased aneuploidy rate was mainly due to our patient age, as most of them were around 40. But even having that issue, we have 3 pregnancies after embryo transfer and 1 live birth, having also 3 more spontaneous pregnancies and 2 live births. In total, the Ascot technique showed a 33% pregnancy rate for very poor prognosis patients with no other option than the egg donation. So, are we inducing any positive long-term effect in patients? If we remind what we have explained, the long-term fertility restoration has been observed in both the diminished ovarian reserve and POI animal model. And some of our poor responders achieve spontaneous pregnancies more than 6 months after the Ascot. In order to analyze that effect, we take samples from the plasma for our patients at the time of the recruitment, during the apheresis, and 3 months after the stem cell treatment. And we analyze the proteome content of the plasma samples by the swab technique. When the 3 conditions, meaning the pre-mobilization, the apheresis, and the post-treatment plasmas were analyzed, we observed that they were clearly different, having done the plasma obtained 3 months after the treatment was still different from the initial condition. We observed that 23% of the proteins in the plasma were differentially expressed 3 months after Ascot. And several of these proteins are related with the aging process and the follicular growth. So in summary, we observed that the Ascot technique for poor responder patients enhances the ovarian reserve biomarkers in 81% of the poor responder patients, increase the antral follicular count 2 weeks after the Ascot, which could mean the rescue of secondary follicles that otherwise will undergo atresia, and it resulted in more follicles, metaphase 2, and embryos developed. although did not modify the embryo quality, because we only have 80% of euploidy rate. But a total of 6 pregnancies and 3 live births were achieved after the Ascot, with 2 women having 2 pregnancies each. But can be the Ascot technique also an alternative for the POI patients? Furthermore, there's another question that should be answered, and is that we have observed no difference in between the infused and the control ovary. So maybe the stem cells are able to reach the ovarian tissue by themselves and to regenerate the ovarian niche, allowing the follicular development. So with all these questions in mind, we designed our next study. This is being developed in POI women diagnosed according to the ESRO criteria, but we have established the limit age in 38 years in order to avoid having patients undergoing our technique, but also having only aneuploid embryos. In this case, we have 2 arms of study. The first one is the Ascot, where we developed the technique exactly on the same way that we did in the POR patients. So the stem cells were mobilized and collected by apheresis and then infused into one ovarian artery. In the second arm, we only performed the mobilization of bone marrow-derived stem cells by a 5-day treatment with granulocyte colony stimulating factors. And in this arm, the stem cells will remain circulating in peripheral blood without cell infusion, in order to see if they were able to reach the ovarian tissue. I'm going to present now some of our preliminary results, as this is still an ongoing study. After the technique, our patients underwent a follow-up of 6 months for the ovarian reserve biomarkers, as well as the FSH levels and the menses recovery, and when possible or follicular growth detected, IVF was performed. To date, we have included 10 POI women. 6 of them were randomized to the Ascot study group and 4 of them to the mobilization. In this table, we are showing some of the basic characteristics of our patients. As you can see, they are characterized by a very high FSH levels and more than a year of amenorrhea. Our preliminary results showed that some of the menopausal symptoms have improved in both arms. In fact, menses recovery has been detected in 33% of the patients randomized to the Ascot study group and 50% of the patients randomized into the mobilization one. And regarding follicular development, we have observed follicular development through several cycles in 66% of the patients in the Ascot arm and 50% on the mobilization one. We have started ovarian stimulation protocols in both arms, and we have patients with metaphase 2 and embryos also in both arms. For now, we have performed 1 embryo transfer in each study group, and we have 1 healthy and 30-week ongoing pregnancy rate in the Ascot group. So these are preliminary data, but we have some take-home messages to give to all of you. Antral follicles have been monitored after treatment in both arms, allowing IVF cycles even by only stem cell mobilization. This follicular growth has been mainly detected from month 3 to 5 after the Ascot, so this can be related with the rescue of primordial or primary follicles. Ovarian stimulation has been initiated in 60% of POI women included, and embryos were obtained in 2 of them. Embryo transfer has been performed in both groups, with 1 pregnancy in the Ascot arm. FSH level significant decrease has been not recorded, but 3 of our 10 patients show FSH levels under 25 international unit at the end of the follow-up. So maybe we should increase this follow-up period to allow the primordial follicles to reach the antral stage and be detected. For now, we should stay cautious and keep researching. I would like to thank to all people from EVRMA, EV Foundation, Hospital Lafayette, and Stanford University who has collaborated with us in this study. And thank you very much for all of you.
Good morning, everyone. First of all, I have to say that I'm happy to be here talking about this topic. I'm going to talk about mitochondria as a tool for oocyte rejuvenation. And in the next 15 minutes, I will point out three important aspects. The first one is that mitochondria are essential for oocyte and embryo competence. Secondly, advanced maternal age, among other infertility-related factors, is associated with a decrease in the function of these mitochondria. And finally, I will show you the different techniques or different approaches that have been proposed for overcoming this situation. So mitochondria are essential, are the battery for the living because they produce energy for the cells. In fact, as you can see in the inner cell membrane of the mitochondria, there is a respiratory chain complex in which ATP is produced at a steady state. Moreover, mitochondria are different because they have their own DNA that is double-stranded and circular. It encodes for 37 genes, most of them related with the function of this respiratory chain complex, and it is much more prone to damage than the nuclear DNA. And finally, it is maternally inherited. That means that most of the mitochondria of the embryo come from the mother. And it is important for you to know that the oocyte has the largest amount of mitochondria and mitochondrial DNA than any other cell in the body, even more than cells with high energy requirements such as neurons or muscles. And it is also important to know that with the age, there is a decrease in the number of mitochondria. And not only this, there is also an increase in the mitochondrial DNA deletions, and there are different morphological changes. And all of them together explain why there is an altered function. In fact, the amount of ATP produced decreases, and there is an abnormal increase of production of reactive oxygen species that lead to an oxidative stress situation. So we could say that the cell more negatively affected by the age is the oocyte, and this aging process explains the mitochondrial dysfunction that could explain also the decrease in the success rates when we are doing IVF because of different factors. So the next question is, can we increase the energy of the oocyte? Can we increase the amount or the production of this mitochondria? Well, different approaches have been proposed. Among all of them, different lifestyle factors have been tested mainly for mitochondrial diseases related with systemic diseases more than with reproductive problems. There are also different nutrients that have been proposed to boost the mitochondrial content, and among all of them, CoQ10 seems to be a promising one, mainly because, as you can see again in this respiratory chain complex, it is CoQ10, coenzyme Q10, it is involved in the production of ATP. And it has been seen that the amount of CoQ10 decreases with the age, mainly in women over 40 years, and it is also decreased in women with background of recurrent miscarriage. And most of the studies have been conducted in animals, but some research conducted in humans has suggested that there could be a benefit by treating patients with CoQ10, but we could say that we need still more research on that to say that it is an efficient treatment for boosting the mitochondria in the oocyte. So I'm going to focus on these last techniques, mitochondrial transfer and replacement, and I will explain you the difference between both of them. To make it simple, mitochondrial transfer means that we are going to inject healthy mitos into the cytoplasm of the H oocyte. The origin of these mitos could be heterologous with the partial cytoplasm transfer or autologous with the germline mitochondrial transfer, the so-called AUGMENT technique. And we have also the mitochondrial replacement therapies that consist on replacing the mitochondrial DNA content, nuclear content, of the donor oocyte by the nuclear content of the patient. They are also called nuclear transfer techniques. And obviously they are heterologous because we are using the mitochondria from a different person, from a donor. We can transfer either germinal vesicle, maternal spindle, polar body, or pronuclear. Let's start with the partial cytoplasm transfer. This technique was tested in the 90s by Jack Cohen, and it consisted on taking a small amount of cytoplasm of a donor, around 5 to 15% of the cytoplasm, and injected it along with the sperm into the cytoplasm of the patient, of the H women. The first results were very promising because they could see a beneficial effect on embryo development and a significant improvement in success rates. But they also realized that there was a high risk of heteroplasmy. That means that you are mixing the mitochondria from the donor and from the patient, and different aneuploidies were described. So finally, this technique was banned by the FDA in the US, and it was no longer used. And this is why this new technique was proposed, the germline mitochondrial transfer, that was trying to avoid the heteroplasmy because it was using autologous mitochondria from the same patient. And the basis for this technique was the fact that some studies in humans indicated the presence of egg precursor cells in the ovary, which could be an autologous source of healthy mitochondria. This technique was called AUGMENT and was launched by the company Ovascience in the year 2014. In summary, it consists on taking a piece of ovarian cortex of the patient and then isolate the egg precursor cells that are present in this cortex. And from them, they obtain the mitochondria. And these mitochondria are injected along with the sperm at the moment of the ICSI procedure. The FDA, again, did not approve this technique, so this is why it was not implemented or not used in the US, and it was mostly tested in other countries such as Canada, Turkey, or United Arab Emirates. In fact, these three countries reported some cases in which they used the AUGMENT technique, and they published their results in 2 papers. The problems were that they did not do randomized control trials, only descriptive or prospective cohort trials. They claimed that there was an improvement in the ongoing pregnancy rate or labor rate, but the control group was the same patient, I mean the historical control, that obviously was a negative result. There was also a difference in egg allocation towards AUGMENT, and the embryologist was not blinded to choose the embryo for transfer, and there was a trend towards transferring embryos coming from AUGMENT group. So this is why in our center we question ourselves if when conducting a randomized control trial, we could demonstrate that autologous mitochondrial transfer could show a better outcome in these poor prognosis IVF patients. We did a study that was published at the end of 2018 in Fertility and Sterility. It was a triple-blinded randomized control experimental study. And it was funded by Ovascience, the company that launched this technique. Our primary endpoint was the ongoing pregnancy rate in the AUGMENT versus control group, and as secondary endpoints, we wanted to compare the embryo quality regarding morphology, morphokinetic, PGTA, and quantification of mitochondrial DNA. We included patients younger than 42 years old, non-obese, with an adequate ovarian reserve and a history of at least one failed IVF due to bad embryo quality. And we excluded patients with any contraindication for the biopsy or very severe male factor. And here you can see the general scheme of the whole procedure that was conducted in our center. The step one was the ovarian biopsy that was done by me, and it was really easy. I mean, it takes not so long to take the sample of the ovarian cortex. We took like 3 small pieces of 6 millimeters, and then we send this tissue to the Ovascience lab that was set up by Ovascience during the study. They isolated the egg precursor cells and obtained the mitochondria. And then we did an IVF cycle, and the day of the pickup, we splitted the eggs of the same patient to receive either ICSI or AUGMENT. I mean, 50% of the oocytes of the same patient were in the control group, while 50% were receiving the AUGMENT technique. In that way, we tried to do an intra-patient comparison to be able to compare the quality in the 2 groups in the same patient. And finally, the step 4 was evaluate the different parameters of embryo quality. And finally, we did a blinded embryo transfer, so neither the patient, the doctor, or the embryologist were aware of the origin of that embryo that was selected according to morphology. Well, here you can see the results. We included 59 patients, although the initial sample size was higher. Why? Because we did an interim analysis. We decided to do this because it was unethical to continue with the study if it was not useful, mainly because it was an invasive technique and not cheap. So we did this interim analysis. In 59 patients with a mean age of 36, normal BMI, a mean of 2.5 previous IVF cycles, and a good ovarian reserve. So the problem was not the quantity, the problem was the quality of these oocytes. As you can see here, the number of meta phase 2 inseminated was 250 in the control, 253 in the AUGMENT group. No differences in fertilization rate, but the first important finding was that the blastulation rate per zygote was significantly lower in the AUGMENT group in comparison with the control group. Moreover, the mitochondrial DNA content in the euploid blastocyst was not different among the euploid embryos and between both groups, even having introduced mitochondria in the AUGMENT group. And the euploid rate was not significantly different, although it was lower in the AUGMENT group. But the most important and relevant finding from the clinical point of view was that the absolute number of euploid blastocysts that we obtained in the control group was 37, whereas it was 19 in the AUGMENT group. And you know that we started with the same number of metaphase 2 oocytes. There were no differences in the cumulative live birth rate per embryo transfer between embryos coming from both groups. So we could conclude that AUGMENT did not seem to improve oocyte and embryo quality in these poor prognosis patients. we decided to end up with a study, and the technique was not implemented in our clinics because we decided to do this type of studies before deciding to implement a technique in our routine practice. Obviously, there are reasons for caution because the isolation of F precursor cells and mitochondrial extraction was performed in a separated lab set up by the company, and several independent groups questioned the reliability of an antibody-based method that is used to isolate F precursor cells from the ovaries of adult humans. Well, and I'm not going to enter into detail into the mitochondrial replacement therapies because there are another 2 lectures in this Congress talking about this, so they will explain you much better the technique itself and the experience that they have using these techniques. But just to point out that the 2 techniques that have been tested for treating infertility problems have been the ones that you see in red, the maternal spindle transfer and the pronuclear transfer. So as far as I know, these have been the 2 techniques that are nowadays under a study to see the effectiveness of these techniques on improving the quality of the oocytes of our patients. I'm not talking about preventing mitochondrial diseases that are another scope. So in summary, these 4 techniques are the ones that have been tested for treating infertility and for improving oocyte and embryo quality. But I would like to remark that the only technique that has been tested with a randomized controlled trial is this one, the AUGMENT technique. And in this case, unfortunately, we couldn't see a real benefit by treating patients with this technique. So I think that it is very important to do this type of research, to do intra-patient comparison, and to analyze very deeply the real benefit of using this type of techniques, because they are not so easy to do, to see if we are able to improve the quality of the oocytes by increasing the amount of mitochondria in the eggs of the patient. And I would like to finish saying thank you to all the team that was involved in this study, because it was a really difficult study, but well, I think that with this wonderful team, we could do a beautiful work. Thank you so much for your attendance.
So I'd like to thank the organisers for making these sessions possible during what is a very difficult time for everyone. And I'm going to be speaking about germline stem cells in the adult human ovary. So we know that the germ cells within the ovary are formed before birth. And that's the primordial germ cells that migrate to the gonadal ridge, and they're forming associations with somatic cells, forming nests, and then the nest breaks down, and then the oocytes will enter meiosis. They will go to prophase 1 of meiosis, the dictate stage, and then they're arrested. And their association with somatic cells forms the structure, the fundamental developmental unit of the mammalian ovary is the primordial follicle. And these primordial follicles are in a kind of suspended animation until something tells them to start to grow. So after birth, there's a fixed population of these primordial follicles, and these must last the female throughout her reproductive lifespan. And these are the regulation of the utilization of these primordial follicles is quite a complex process, but follicles start to grow continuously throughout life, even before puberty. So the oocyte primordial follicles will start to grow, the oocyte will increase in size and it's acquiring its developmental competence.
the somatic cells, the granulosa cells, are proliferating, and then we get the formation of the large antral follicle, and finally the pre-ovulatory follicle that consists of the oocyte surrounded by the cumulus cells and then the murogranulosa cells. Now, we've always thought that the mammalian ovary has no capacity for regeneration. And of course, we know that in humans that you run out of eggs and that leads to the menopause. But recently, there was work from Jonathan Tilley's lab, and at that time he was in Mass General Hospital in Boston, part of Harvard, and he had discovered or had shown that he could isolate a population of cells from the adult human ovary that had a marker for germ cells, DDX4, an RNA helicase, and these cells also proliferated. They picked up BRDU. So he concluded that these cells were a population of cells that could lead to the formation of new oocytes. And he did some elegant experiments where he marked these cells with green fluorescent protein, and he injected them, he isolated them, marked them with the green fluorescent protein, and injected them into some human ovarian cortex, and then transplanted them into an immunodeficient mouse. And what he obtained was primordial follicles or growing follicles where the oocyte was green, so indicating that these oocytes had been derived from the cells that he had injected into the cortex. So this was a really interesting work, and prior to that, I and other members of my group and many scientists were very skeptical about the existence of cells within the adult human ovary that could form new oocytes. But John Tilley, at that time, shortly after he published this paper, was in touch with our group because we had been working on this multi-step culture system to support the growth of primordial follicles from the human ovarian cortex, where we had this process where we could get them to activate to grow, we could isolate growing follicles, and then we could obtain fully developmentally competent oocytes all the way from primordial follicles. And he asked, well, would we inject some of his cells into this system? So we used this multi-step culture to look at these putative oogonial stem cells. And what we found was using these cells was when we injected them, you can see the green fluorescent protein, there's cells that are injected into the cortex, and within a few days, we started to see clusters forming. And when we looked after 7 days and we did some immunocytochemistry, so we looked at the fluorescent marker, we could see cells that had been injected in, and we could see structures that were larger. But of course, we couldn't really see much detail, so we had to use a chromogenic marker to look for the GFP. So when we did the immunohistochemistry and we were looking for GFP cells within the ovarian cortex, we could see different stages of development. So we could see what we refer to as type 1, so small cells that look like the cells that were injected in, and then type 2, where these cells are becoming larger and they're forming associations with somatic cells, the somatic cells that are already in the ovarian cortex. And then you can see here type 3, it looks like they're forming follicle structures. And then type 4, and this is very, very much like an ovarian follicle, a primordial follicle. So we were convinced then that these cells did have the capacity to form what appeared to be new oocytes. And these were cells that, these initial experiments were cells that had been isolated in Tilley's lab. And he concluded that these cells were unipotent progenitor cells, so cells that would only go down the route of differentiation towards oocytes. Now, this was heavily criticised, the work that Tilley published, and not only Tilley, but there were other groups in China and Italy and in Slovenia. And these methods were based entirely using an antibody to the RNA helicase DDX4. The other point about the work that Tilly had published was the only cells that had been cultured, they'd been cultured for quite some time, around 3 months. They were the only cells that had been tested. So the argument was that there was potential for in vitro transformation. So we wanted to independently isolate these cells and see how reproducible this technique was, but also to ask the question whether you took freshly isolated cells, would they have the potential to form oocyte-like structures? So we used a similar method, and one big difference that our work has in comparison to other groups is the method that we use to dissociate the cells. The published dissociation method was very harsh, and so you had the danger of losing the cells of interest. So we worked out a methodology to ensure that we would not kill these cells. So it was a less harsh dissociation method. And we used the DDX4 antibody as a marker, but we also used aldehyde dehydrogenase. And aldehyde dehydrogenase, using this substance aldofluor, it detects aldehyde dehydrogenase within the cell. And this is a marker of stem cell potential of a cell. So stem cells have quite a high level of aldehyde dehydrogenase. And the fluorescence that we find if aldehyde dehydrogenase is present is only found within intact cells. So we don't see it in degenerating or the dead cells. So it's quite a useful marker to look for viable stem cells. So we analyzed, we sorted our populations of cells according to DDX4 intensity and aldofluor intensity. And we concluded that we could find probably 3 populations, and there may well be more, but we've categorized them into their DDX4 and the aldofluor intensity. So when we look at the markers, so some of the germline markers and stem cell markers, we find that these populations express germline markers and stem cell markers, whereas the negative cells that don't have DDX4 or the aldofluor, they don't have these markers. But interestingly, we found that in our population 2, we could see DASO, which is a precursor to the cells entering meiosis. So we conclude that perhaps we have sort of different stages of development of these cells, but we need to refine our isolation methodologies to really characterize each of the subpopulations. Another thing that we observed was that we, in population 2, we found a splice variant of the DDX4. And DDX4 has many splice variants. So we concluded then that we could isolate cells that had germline and stem cell markers, but these cells were not oocytes. And what we did was we took these positive cells, these putative germline stem cells, and mixed them with fetal somatic cells. And we found that we could form these aggregations. And when we looked at the histology of these aggregations within 2 weeks of the culture, we could find that there was some structures that looked very, very much like primordial follicles or early growing follicles. So these cells seem to be forming structures that were indeed oocyte-like. And we could see some larger structures as well after, up to 21 days in culture, we were seeing these larger structures. So what we've been doing with this is, of course, we don't really know if these are true stem cells or they're cells that, when they're put in certain conditions, will form what appear to be oocytes. But what we're working on now is how we can optimize the development of these cells by looking at the somatic cell progenitors. So looking at somatic cells that will support the growth and development of these putative germline stem cells. And at the moment, we're just looking within the DDX4 negative population within the ovary, and we're trying to refine this population. But when we combine these cells with the putative germline stem cells, we find that we get aggregates, and when we look in detail at these aggregates, we find that we have what appear to be follicle-like structures and oocytes formed, or oocyte-like structures. And when we analyze these oocytes, we can remove these oocyte sort of cumulus type complexes, so from the in vitro grown cells that we've derived from the oligonuclear stem cells, or putative oligonuclear stem cells, we find that they do in fact express markers that we find within the oocyte. So they're positive for GDF9, indicating that they are going down the differentiation pathway towards becoming oocytes. So again, these are preliminary works, but I think it shows the potential of these cells. So in summary then, distinct populations of cells with germ and stem cell markers can be isolated from the ovaries of adult women. And by combining these cells with an in vitro growth system, and we've developed a multi-step system, then that provides a useful model to study human germ cell development, because we really still don't understand so much about human germ cell development. We're now characterizing the subpopulation that we can find within the adult human ovary, and we're looking for the optimal somatic cell support to complete oocyte development. But we need to concentrate on defining the populations of cells that reside within the mature ovary, and to determine their potential both in vitro and in vivo. Now, there is still a great deal of controversy surrounding this area, and there's papers, in fact, a recent paper has been published that emphatically states that there are no germline stem cells within the adult human ovary. Well, within that paper, there's also a number of other cells that they don't find within the adult human ovary. And of course, we do know that ovarian surface epithelial cells are within the ovary, and a whole range of cells. So it's difficult if your technical issues are maybe destroying the cell population that you're trying to find. You cannot really state that they're not there. But if we think about it, it's likely that the ovary has stem cells for all of its components, because we now know through the fast developing field of adult stem cells that almost every single tissue that has been looked does indeed have stem cells. The ovary is probably not that different, but that doesn't mean that they're actively involved in forming new follicles or oocytes during normal physiological conditions, but they may well be there to form new eggs under perturbed conditions. So I think we do have to continue to look at this cell population within the human ovary. So what does it mean for patients? Well, it means that cells that have potential to form new human oocytes in vitro can be isolated from the cortex of adult women. There are, of course, potential clinical applications in these isolated cells, particularly in fertility preservation and restoration. But we need to test the developmental potential of them in vitro derived oocytes, and we need to ensure that they're safe, and that's ongoing work by a number of groups across the world now. We're still a long way from application, but future planning should be considered when women are storing ovarian tissue, that to see or to determine whether these cells can be isolated and stored is something that we should look to the future. So I'd like to thank everyone for your attention, and to thank the people who do the work within our lab, and of course my clinical collaborators, but mainly Richard Anderson has been involved in this project, the funding that we get from the Medical Research Council, and of course patients who kindly donate their ovarian tissue, and the clinical teams that support our work. Thank you very much.
Yeah, first of all, I'd like to thank the committee for inviting me to be this very unique opportunity. Here, I'd like to talk about our study in vitro activation of follicles for infertility treatment in poor ovarian response. This is my COI. So here, and in the beginning, I'd like to mention about premature ovarian insufficiency, POI. Diagnosed by amenorrhea before 40 years of age and hypergonadotropic hypogonadism. So these patients showed infertility and symptoms caused by estrogen deficiency, such as hot flushes, mood disturbances, sexual dysfunction, etc. In terms of etiology, POI affects approximately 1% of women. Specific features of POI lack in follicle growth and ovulation, and exhaustion of the ovarian follicles, and thus, if we try to measure the MH level, almost all the patients showed undetectable levels. And these patients should have very limited residual follicles, should be less than 1,000, and because of these specific features, the patients are really resistant to traditional gonadotropin treatments. And egg donation is the most successful treatment option, but the patient, the couples would like to have their own genetic babies. Because even in the case of the POI, they still have some residual follicles in their ovaries. So we hypothesize, is it possible to activate residual dormant follicles in POI patients to develop a new treatment? Primordial follicles are dormant in ovaries. Some growth factors such as kidligand, neurotropins, BMPs, BGF, REEF have shown to be activated from primordial to primary stage. Among huge number of dormant follicles, up to 0.1% follicles selected and activated per cycle. Because the PI has less than 1,000 residual follicles, so that means theoretically, these growth factors can activate only one follicle in those cases. So it's almost impossible to use a clinical setting. So that's why we focus on an intracellular signaling system involving the activation of primordial follicles. Based on knockout mouse studies, Reddy and Casrilon reported similar phenotype in P10 knockout and HOX3 knockout mice. So as you can see, at day 35 after birth in the wild-type animals, there's a follicular activity, and P10 knockout mice, it looks like totally different. Ovary became bigger, and you can see multiple growing follicles inside. They are originated from the spontaneous activation of primordial follicles. And at 6 weeks after birth, in the wild-type animals, they still have a follicular activity. You can see the different sizes of follicles, including the corpus luteum. But in the P10 knockout, the ovary became shrunken, and almost all the follicles are depleted. And completely similar phenotype could be obtained in a HOX3 knockout mice. Okay, so this similar phenotype, this similar phenotype could be explained by this PI3K signaling pathway. This pathway begins the PI3K activation by receptor tyrosine kinases after binding growth factors. And then PI3K activate AKT, which inhibit the activity of FOXP3, resulting in cell proliferation and survival. This molecule, P10, negatively regulates this PI3K signaling pathway. So in primordial follicles, local factors activate dormant follicles through this pathway, whereas the P10 acts to block the signaling. Then again, we have that is it possible to activate residual dormant follicle in POI patient artificially by transient PTEN suppression and or PI3K activation using some kind of drugs. We have a PTEN inhibitor, we have a PI3K activator. Using these drugs, we assess our hypothesis by animal studies and confirmed treating with an ovaries with this PTEN inhibitor and PI3K activator can activate dormant primordial follicles. Then we move on the clinical study. This is a brief scheme of our clinical study. The first step is ovariectomy under laparoscopic surgery. And then we culture ovarian cortical tissues with PI3K activators for 2 days. So as an option, we can prepare the ovarian cortical stripes for freezing. Now we are using a vitrification method for cryopreservation. And during cryopreservation, we can do the histological analysis to detect the residual follicle in the ovaries. And then after 2 days culture, we transfer the ovarian cubes to the patient again. And then after that, we can do regular in vitro fertilization embryo transfer treatment. So here, I dissected ovarian cortices containing residual follicles by removing the medulla and cut into small stripes around 1 centimeter square with 1 to 2 meter thickness where residual follicles are located. And as an option at this moment, we can do the cryopreservation by vitrification method, as I mentioned. And usually around 6 to 8 pieces of ovarian stripes could be obtained from one pure ovary. And using 10% of volume each ovarian stripe, we are doing a histological analysis to detect the residual follicles. And then ovarian stripes further fragment into very tiny 1 to 2 millimeter square cubes here, and put onto the membrane of the cell culture insert and covered by a very thin layer of the media, including a PTEN inhibitor and a PI3K activator for 2 days to activate dormant follicles. Then this slide shows the odd transplantation of ovarian cubes. Under laparoscopic surgery, I carried a serosa and made a pouch between the serosa and the fallopian tubes here, and then grafted multiple ovarian cubes beneath the serosa of the fallopian tube. And then the wound was covered by the oxygenized regeneration cellulose to avoid the cube loss from the graft site. Of course, you can make a suture instead of covered by this oxygenized regeneration cellulose C. Then we succeeded to get a baby. It was published in 2013, and now we are getting more and more babies, and also the other countries like China and Spain and so many other countries could repeat our success of this IVF treatment. And here I'd like to show the temporal follicle growth in transplanted ovaries. Because it takes around 4 to 6 months from primordial to preoperative stage in a human, so first we expect to have a follicle growth after 4 to 6 months of the grafting surgery. But in contrast, we could get a follicle growth, not all the cases, but in some cases, we could get a follicle growth before 4 to 6 months after grafting. So these results suggest our IVF method also stimulated growth of secondary follicle in grafting ovaries. But the issue is, question is, how could activate dormant primordial follicles grow into preoperative follicle in just a few weeks? So I talked about the possibility of the activation of the secondary follicles. So then the question is, are we unleashing the growth potential of arresting the secondary and preantral follicles after ovarian fragmentation in a PI patient? What is the molecular mechanism? So we tested our hypothesis by cutting mouse ovaries at day 10 stage that include both primordial and primary and secondary follicles. And one side is intact ovary, the other side cut into 3 pieces and grafted to the kidney capsule of recipient mice. And after 5 days, we removed the grafting ovaries and weight their measure their weight. And as you can see, the ovaries cut into 3 pieces became heavier than the intact ovary. And then we checked the histology. So here you can see the proportion of the late secondary and antron preorbital follicles are increased in the group of the ovaries cut into 3 pieces, and a little decline the proportion of primordial follicle in the group of the 3 cut into 3 pieces. So this result suggests ovarian tissue cutting the tiny pieces, cutting fragmentation is really increasing, stimulating the secondary follicle growth. But again, the question is, what is the molecular mechanism behind? Because of the shortage of time presentation, I cannot show the detailed data, but here I summarize our findings. So here, the ovarian fragmentation led to the change in intracellular tension and facilitated the conversion of the globular actin to filamentous actin. And then this filamentous actin suppressed the hyposignaling pathway, allow YAP going to nuclei, and this YAP worked with CCN TAD translational factor to produce CCN growth factors and anti-apoptotic factors. And these factors can stimulate the secondary follicle growth. So here, now you know, our in vitro activation IVF approach contains 2-step follicular stimulation. One is a PI3K signal stimulation to allow to activate the dormant primordial follicle to be primary stage. The other one is a hyposignaling disruption. This signal changes allow the secondary follicle to be an antral stage. And then here I'd like to mention about the poor ovarian response patient. Here the Bologna criteria, very well known the criteria, and at least 2 of the following 3 features. One is advanced maternal age or any other risk factor for POR. And the second is a previous POR, less than 3 oocyte with conventional stimulation protocol. The third is an abnormal ovarian reserve test. For example, antral follicle count less than 5 to 7 follicles, or MH levels was is less than 0.5, 1.1 nanogram per ml. So etiology of these PR patients, around 10% of women undergoing an IVF. So here's a recent Poseidon certification. So we are focused on the group 3 and group 4 patient. This patient is like a diminishing ovarian reserve. Usually people call it ovarian dysfunction. In this type of the patients, actually, this is the image of the antral follicle count and MH levels. So here, in the beginning, I mentioned about the POI patient. They have only limited number of the primordial or primary follicles in the severe cases. But early stage of POI, they may still have a secondary follicles. And here's a PR patient with diminishing ovarian reserve. They have much higher number of the secondary and even early antral follicles, but they could not detect any antral follicles, or very limited number of antral follicles. Of course, their MS level is very low. So we try to develop a new IVF approach targeting immature secondary follicle in the case of the patient, PR patient with diminishing ovarian reserve and early POI patients. In those cases, without doing the drug treatment, I mean the activation of the dormant primordial follicles, maybe we can do the stimulation of residual secondary follicles just by disruption of the hyposignaling only. So here's our original IVF approach. We require 2 times the surgery, but in the case of the DOR or early PI patient, we just remove the part of ovarian cortex and further fragmented into tiny pieces as shown before, like 1 to 2 millimeter cubes, and immediately we can return to the patient. It requires only 1 surgery. So here I first try to show our surgical approach. We just remove the part ovary like this, and then cut into tiny pieces and grafting back to the residual ovary or the beneath the fallopian tube, like original IVF approach. So in the original IVF approach, we prefer to return to the beneath of the cells of fallopian tube because of very few blood circulation in the remaining ovary. But in the case of poor PR patient with reminiscing ovarian reserves, they have better blood circulation in their ovaries. So that's why we return to the both residual ovary and the beneath of the cells of the fallopian tube. And now we are doing a clinical study. And here, the limitation of this new approach is only applicable to the DOR early PR patient who are likely to have a secondary follicle, but there are so many advantages. So minimum damage to the ovarian blood supply, no need to use a drug treatment, avoid potential follicle loss during culture, 1 laparoscopic surgery. This is important. And if we try to grafting back to the residual remaining ovary, we can expect spontaneous pregnancy. Actually, EB group in Spain, they reported the success of 3 spontaneous pregnancies out of 14 patients using this IVF, drug-free IVF approach. So here I'd like to share with our clinical data, and we treated 11 patients, and they have this background. The serum FS level was median was 47, estrogen level is 0. MH level was 0.04. And then we have done the drug-free IVA. For a surgical approach, we just try to remove the ovarian cortex, try to avoid to reach the medulla to avoid bleeding, and then cut into tiny pieces. And using this very unique device like a plunger, you can put around 10 to 20 ovarian cubes here and insert in the remaining ovary and push out all the tiny ovarian cubes. And then the wound could be covered by the interseed. Or of course, you can make a suture. And here's the outcome. So this number, right hand, is the mean antral follicle count before the surgery. For example, patient number 2 was only 0.7. That means every time one follicle or 0 follicle grows. And suddenly after the surgery, here's we observed 1 year, and then she could have multiple follicle growth, and this number in the over means how many antral follicle grown at the time, and 6, 8. So it's totally increased. And red color means after the surgery, antral follicles count was increased. So of course, not all the patients, unfortunately, number 3 and number 5 were not responded, but most of the patients were responded to increase the antral follicle count. And then we could have a successful pregnancy in the 4 out of 11 cases, and 3 patients who have cryopreserved embryos are waiting for an embryo transfer. So that means we could have much higher pregnancy rate in future. So we have all, this approach, international patent. And now we are trying to do the more and more patient with these collaborators. And also here, I'd like to thank Professor Aaron Shui in Stanford University for long-term collaboration of establishment of original IVA and drug-free IVA approach. Thank you for your kind attention.
Thank you, IVF Worldwide, to invite me to speak for their first online congress. And thank you to the listeners. I have got no conflict of interest to disclose. UK became the first country in the world to legalize and permit clinical use of assisted reproductive technologies to prevent transmission of mitochondrial DNA disease. This all happened from Newcastle, which is in the northeast of England. As you can see, the red pointer marking towards that. This is where I belong, and I'm giving this talk from. We all are 2G humans. That is, we have two genomes. One is the nuclear genome, which we inherit from our father and mother, and the second one is the nuclear genome, which we inherit only from a maternal line. The nuclear DNA, as we all know, is a double helical structure, but the mitochondrial DNA is a circular DNA. It encodes very small number of genes, that is around 37 genes. And it does, if you look at an egg, there are multiple copies of mitochondrial DNA. More than 90% of our cellular energy comes from these mitochondria. So they are the powerhouses of our energy production. That's why they are crucial, and they provide the optimal health of all our organs and tissues. But one needs to understand that mitochondrial mutation load, if someone has a mitochondrial DNA mutation, is not same in every single egg of that individual. So if a woman has got mitochondrial DNA mutation, some of her eggs will be absolutely normal with no mutation load at all. The green is highlighting the wild type mitochondria. The red one is the mutated mitochondrial DNA mitochondria. If you look at it, there are three types of embryos and eggs shown in this picture. The first one, which is completely normal looking, will result in normal embryos without any mutation load transmitted. However, there could be variation in the amount of mitochondrial load that goes on to the embryo and the resultant child, and this is known as heteroplasmy. As a result of this heteroplasmy, there is a founder effect that based on the mitochondrial DNA mutation load, we can have different types of embryos created, and even each individual embryo may have different amount of mitochondrial DNA mutation load going to different organ systems. So all the three germ layers can be affected by this, but based on how much mutation load is in that particular tissue, it may be different manifestation of the severity of the disease. So as you can see, mitochondrial DNA mutation can cause disease which is neurological based and non-neurological based, but mostly it affects all the high energy requirement organs and tissues. The prevalence as such is known to be around 1 in 5,000 to have mitochondrial DNA mutation. Newcastle is a hub as a supra-regional service which provides for mitochondrial DNA mutation patients and those who have this disease. So they are looked after here, and we get referral from all across the country. This is one such family where the mum was affected with mitochondrial DNA mutation disease. Her load was only around 38%. She herself was clinically well. She had 2 kids. Her son had 78% mutation load, whereas the daughter had only no load at all, which was very healthy and fine. Unfortunately, her son died at the age of 7 with multi-organ failure. A mum like her, can we help such moms who have living in a constant fear of not knowing whether they will have a child who may be affected and whether they will be able to see the child go through life as normal as someone else, or their child may not even go past the primary school? That made us in Newcastle think about, can we help these patients, these women and families affected by this condition to minimize the risk of inheritance of mitochondrial disorders using assisted reproductive technologies? So far, there is no known cure. So is there anything else we could do? What are the current reproductive options for reducing the risk of transmitting mitochondrial DNA mutation? What we know is that pre-conception, one could do PGD, which is pre-implantation genetic diagnosis, and pick up the embryos which have got the least mutation load. Or post-conception, if someone has conceived naturally or so, they could have prenatal diagnosis. In between of this is the option where we don't use the woman's eggs at all and go for egg donation. If you're using prenatal diagnosis, you could take some cells out from a growing embryo or the cells around it and test it. However, if we just focus on PGD for a second, the talk is mainly for pronuclear transfer, which I'll come to, but PGD, we think that While it's a promising risk reduction strategy for affected families and in assisted reproductive technology field, we are used to doing PGD treatment cycles. However, it is not appropriate for everyone. We can't offer this to everyone, especially not for women who are homoplasmic for this mitochondrial DNA disease, or for women who are heteroplasmic, but they consistently produce embryos with significantly high mutation loads, guaranteeing that those babies will be affected when they are born, and they will pass it on as well if they live up to that age. So the question that came to our mind was that, can we uncouple the inheritance of the nuclear DNA and the mitochondrial DNA? And can we just transfer the nuclear DNA, which is fine, so that the genetic identity is maintained, but reduce the transmission of the mitochondrial DNA by just putting healthy mitochondria inside? And the answer to this was, even though we did not have a cure before, this promising new technology, that is mitochondrial replacement therapy, gave us a hope that we will be able to detect it early, fix it early, and prevent the transmission of mitochondrial DNA disease, not only for the child who will be born from this treatment, but also for future generations to come. So this is what we wanted to do, so that the families did not have to play a genetic gamble of whether the child will be normal or not. But we didn't know, will it work, or is it just a therapeutic illusion that we are thinking, oh, maybe it will work, and we don't know yet. So we needed to get into the science and technology of this new visionary idea that we wanted to explore. There are 3 types of technique in mitochondrial replacement therapy, and I'm not going to cover on the spindle transfer or polar body transfer, which would have been covered by the previous speakers, but I'm going to focus on pronuclear transfer technique. All these 3 techniques, although done differently, result in a reconstituted zygote with different mitochondria from someone else and the nuclear DNA from the recipient, from the mother who is affected by the condition. So addressing the science, we had 3 main questions to ask. Will it work? Is it efficient? And will it be safe? And 2 key scientists I would like to highlight here is Professor Mary Herbert from our lab, who led this research scientifically, and Louis Hislop, who is our embryologist, who conducted all the scientific experiments in our lab. However, to start the research, the first challenge we faced was the research material. There was no ready supply of normally fertilized eggs available for research. No woman who's going through IVF treatment would donate their normally fertilized eggs just like that for research purposes. Therefore, we initially decided, let's try the proof of concept experiment by using abnormally fertilized eggs which were not suitable for women's own fertility treatment. However, there was a regulatory challenge that we needed to address. As per the HFEA Act of 1990, which is governed in UK, it stated that the license does not authorize altering the genetic structure of any cell as long as it forms part of an embryo. Now, performing pronuclear transfer, would it mean that we are altering the genetic structure? That was a key question which was debated in the HFEA and public consultation extensively. And it was concluded that pronuclear transfer would generate new combinations and conglomeration of the nuclear and mitochondrial DNA, but we were not altering the genetic structure, so it should be allowed. And hence the HFEA granted us research permission to do this technique. So we did the research. This video is going to just demonstrate to you the technical challenges we faced and doing the technique. So you can see the 2 pronuclears in this zygote, and you can see a pipette going in to taking out the pronuclears. Initially, we tried taking both pronuclears out together in one setting. However, we realized that it was getting a lot of cytoplasm with it, resulting in a high mitochondrial DNA carryover. So we started using that pinching technique, which you just saw, where we pinched the cytoplasm away and took 1 pronuclear at a time rather than removing both pronuclei together. So this is the second pronucleus now been taken out, as you can see. Once we took out both the pronuclei, the next step was to expose this karyoplast, this pronuclei called as karyoplast, to HVJ viral protein to facilitate fusion when they're mixed with the donor zygote. So this is an enucleated zygote where the pronuclei has already been taken out. And you can see we are replacing the 2 pronuclei from the recipient zygote into this. And slowly we are injecting it into the egg. And then afterwards, you would love this technique of Louis, where she's giving the nudge a little nudge to the embryo so that the embryo can go on to develop into a blastocyst. So this was a proof of a concept experiment using abnormally fertilized eggs, although the video was of a normally fertilized embryo with 2 pronuclei. That was done later on when we started those experiments. And with the proof of concept experiment, we showed that it was technically feasible. It was compatible with development to blastocyst stage. However, if you look at this graph and the numbers here, you can see unmanipulated controls of abnormally fertilized embryos. The blastocyst formation rate is lower at around 16–17%. And with the PNT technique, those embryos from the abnormally fertilized eggs did result in blastocyst formation, but the rate was significantly low at around 8%. That's really, really low. But at least it showed that it was compatible with development to blastocyst stage. And also when we looked at the mitochondrial DNA carryover, it was noted to be low. However, to take it to the next step to clinical translation, we wanted to do meaningful tests of safety and efficacy, and that required a supply of normally fertilized eggs. So the next challenge was to get these normally fertilized eggs. Where could we get it from? So we applied for HFA research approval as well as ethics committee approval. We started off with egg sharing for research scheme, where women who were undergoing IVF treatment opted to participate in research program and donate half of their eggs for research. The treatment cost was subsidized in benefits in kind system by the research funds, and it was subsidized by £1,500. And this I published in Cell Stem Cell Journal. However, we noticed that although it did provide us with some eggs, it was not sufficient for the research experiments that we were planning to do. Then next step was to recruit healthy volunteers who were not participating in any fertility treatment, but non-patient donors who donated eggs for this research project. Currently, around 80% of our research oocytes come from this source. They were compensated as per the HFA approval permitted following 2011 review of £500 per donation cycle. This has significantly increased the number of eggs which are available for research. And this would not have been possible without the 2 landmark reports, one from the HFA with the new policy on compensation and one by Nuffield Council on bioethics looking into donating for research purposes. However, although the compensation policy may suggest that, okay, a lot of women will come forward and donate it, what we found is that in Newcastle, despite several women expressing interest to participate initially, only 5% eventually went on to donate their eggs, which was a significant finding actually. So looking at normally pronuclear transfer using normally fertilized zygotes by these 2 sources, we did the process at around 16 to 20 hours post-insemination of the eggs with the sperm to replace the pronuclear transfer, but we had a huge setback. 40% of the eggs that were fertilized and we did PNT on did not survive. Now, we cannot afford a patient going through treatment to lose 40% of her zygotes in this manner. We were at the end of a dark tunnel. We all in the world globally right now facing this dark tunnel because of the COVID coronavirus pandemic. And we hope there will be light at the end of this tunnel. And we are seeing that light in a lot of countries now as the death toll reduces and the number of cases reduces. Similarly, in our research project, we also noticed light at the end of the tunnel. This is Newcastle on one of the days when we were allowed to go out into the city. And you can see a beautiful light at the quayside with the Millennium Bridge. Hopefully, once the travel ban is lifted, you would all like to visit our beautiful city of Newcastle. But we saw that light at the end of the tunnel and we went back to the drawing board to troubleshoot the problem. What we thought is it possible that there is insufficient recovery time between PNT and the first mitotic division? And if we brought it forward, say around 8 hours post-fertilization rather than 16 to 20 hours post-fertilization that we were doing, would we manage to solve this problem that we were facing of losing 40% of oocytes? And if you look over here, the graph on the right side, the blue panel is for the late PNT process and the purple panel is the early PNT process, and you can see significant improvement in the survival of around 90% in the reconstituted zygote in the early PNT group compared to around 60% in the late PNT group. That was very promising. We also looked at the blastocyst development rate, and if you look at the graph on the right side, you can see that the gray panel denotes control group unmanipulated embryos. The blue one is the autologous PNT zygotes, which means the blastocyst created from that. What we mean by autologous is that 2 different eggs were used to reconstitute the blastocyst, but from the same woman. And the pink panel is from heterologous PNT process, where eggs from 2 different women were used to do the process. We would expect the blastocyst rate would be lower in heterologous group compared to autologous group. And you can see autologous group, it was very comparable to uncontrolled, unmanipulated controls. However, despite a lower blastocyst formation rate in the heterologous group, you can still see it was 30 to 40% compared to 8%, which was in the abnormally fertilized embryos. From the blastocyst, we also wanted to maximize all the possible information. We looked at gene expression using single-cell RNA sequencing. We took some of the cells for pyrosequencing to analyze mitochondrial DNA carryover, as well as we took some cells for aneuploidy screening by RACGH method. We did confirm there was no increase in aneuploidy detected in EPNT blastocysts, which was reassuring, as well as no detectable effect on the expression of genes encoded by both nuclear and mitochondrial genome. So this helped us to conclude that EPNT technique was likely to be compatible with establishment of viable pregnancies. But the key question still remained, will it prevent transmission of mitochondrial DNA disease, what we set out to do in the initial first stage? To understand that, we need to know about 2 things, disease and inheritance prevention. So one is whether it would reduce the transmission of a disease. So based on this paper from 2012, what one understands is that if someone has a mutation load of less than 18%, then there's a low probability of disease symptoms manifesting. Probably they would have either no symptoms at all or very mild symptoms. In short, they would be able to lead a healthy life, more or less. However, to prevent it transmitting to the next generation, the mutation load has to be less than 5% because of the heteroplasmy we discussed. If the low probability of transmission to the next generation will be possible, then we will be able to prevent the disease transmission. So when we did our mitochondrial DNA carryover and we published it in Nature, we found that the mitochondrial DNA carryover was less than 2%. It is very important that we get this as low as possible because one of the 4% mitochondrial DNA carryover embryo, when it was cultured further in the stem cell line, there was repercussion seen where there was a changeover and increase in mitochondrial DNA possibility. So at present, what we can conclude by our experiments is that we can minimize the risk of transmission, but we cannot guarantee that as of yet. So based on all these experiments so far we have discussed, we could prove that it was technically feasible to do this process. It was compatible with onward development of the embryo without genetic compromise, and it has the potential to prevent transmission of mitochondrial DNA disease, but further research is warranted to guarantee a prevention altogether. So we were very happy that we had managed to address the key science questions we had set out to. It was working, it was efficient, and it appeared to be safe. And this resulted in 2 Nature publications from the experiments done from abnormally fertilized and normally fertilized embryos. The next step was to understand the regulatory and ethical aspect. Will it be legal to do it for clinical translation, and will it be ethically acceptable? Research is one thing, but clinical translation is the second thing. It took us 10 years for our research to get from starting the experiments to getting the results. And if we waited that long, it would take another 10 years for the regulatory process. So we decided we'll do it in parallel. However, when we started the process of changing the law and all that, you can see with scientific vigor comes media frenzy and ethical hype. The media started labeling us as Frankenstein's monster. And it was a big hue and cry in the media whether it should be allowed or not, the ethical perspective. But one thing that helped move it further was the Nuffield Council on Bioethics report on ethics in mitochondrial replacement technology. Is it a slippery slope? And they addressed all the key queries, issues that has. The germline therapy, is it a form of a germline therapy? Will it result in any risk to resultant child? The safety and efficacy, it's uncertain at present, but what it is. The mitochondrial donor status, will it be similar to the altruistic donor who donate their whole leg, or will it be different? In UK, the anonymity law is different for both the groups. And lastly, the self-identity conflict. Will any resultant child from this, will they have an identity conflict based on this? They sensibly concluded that if the scientific team can establish that these techniques are safe and effective, on balance, it could be ethical for families wishing to do so to take this opportunity and go ahead and proceed with mitochondrial replacement technique. However, this should be subject to provision of adequate level of information and support, including counseling support. So in parallel with science, we were progressing with the legal and regulatory framework, because as I said, legal and regulatory framework also takes time, and it took almost approximately a similar decade time. If you see, we got the first license in 2005 for the research from the HFEA. And if you look at all this, we managed to do experiments slowly. And every time we did experiments and published the data, HFEA would convene an expert panel to review the scientific evidence to see whether it should be allowed or not. On the regulatory side, the Parliament was debating whether it should be passed or not. And then in 2008, the Parliament gave the provision to allow passing this regulation. However, the regulation was not passed by both the Houses of Parliament till 2015 February, and it came into force in October 2015. Subsequent to that, although the regulation was passed and it was approved and came into force in October 2015, it was another year before the HFEA permitted use in clinical practice. So HFEA invited applications for treatment license in December 2016, and in March 2017, the first license was granted to Newcastle Fertility Centre, and it remains the only centre in UK to do this technique. But it is licensed for only prevention of high risk of transmission of serious mitochondrial DNA disease. It's not allowed for recurrent implantation failure or other indications. So far, HFEA has approved more than 15 license applications to offer mitochondrial donation treatment to us. And we also have secured separate funding for Wellcome Trust for follow-up of the first 25 babies. Unfortunately, I'm not in a position to disclose these results, but I promise you that once we are in a position to disclose and share these findings, we will be able to share the clinical outcome data as well. This is just the beginning. We need to know that, as I said, it's not a guarantee of prevention, so we need to have a long-term follow-up plan as well as ongoing research simultaneously. To conclude, I would like to say that none of this would have been possible without the source, the one who powered the powerhouses, the mitochondrial donors for research, and now the mitochondrial donors for treatment as well. These women who have donated their eggs selflessly for research and for clinical treatment have not only given a hope of giving a healthy child to a mother, but they have also given hopes to millions of future generation of children to come by preventing transmission of this disease. And we are very thankful to them. Clinical translation globally has started with PNT and MST technique for mitochondrial replacement therapies for different indications in different parts of the world. But we all should aim for a common goal that it should be safe, it should be ethical, legal, reproducible, and it is done for the right reasons. And that's what we all should come together as a scientific community and clinical community and aim for. And this we owe to our patients and the future generation. And with that, I would like to conclude my presentation and thank you for all listening. My email address is mentioned here. If anyone wanted to get in touch with me, I would be happy to address any questions or for networking reasons. Thank you so much.
Thank you, Mina, for your interesting presentation. I would like to apologize to the speaker and the audience for not having questions and answers in this session. Time is short, and all the questions will be sent directly to each of the speakers. We therefore ask Evelyn to summarize the 5 presentations of this session. Please, Evelyn, please summarize it and go ahead.
Okay, thank you. So the first presentation was by Sonia Herrales and Evie in Valencia. And she presented a very nice study where they were to recover the ovarian function in POI patients using treatments to mobilize bone marrow stem cells. And they could mobilize the stem cells using a granulocyte colony stimulating factor. And in one group, they would collect the cells and then infuse them back into the ovary. And then in the other group, they had just mobilized the cells. In both treatment groups, they had a positive effect on the growth and development of follicles. And Sonia is now looking at improving these treatments to mobilize or activate primordial follicles and facilitate growth in patients where there are fewer follicles. In the second presentation, Dr. Elena Labata from EV in Valencia looked at her experience, or their experience of AUGMENT, and that's injection of autologous mitochondria from these egg precursor cells to improve the outcome in patients who had previously failed IVF. Now, the results presented here indicate that this technique doesn't result in improvement. So indeed, whilst this idea sounds very promising, there's actually been several studies worldwide where they've been unable to provide robust clinical data that support this really innovative idea. And then in the third presentation, it was my presentation, and I discussed results from my lab that showed that there are populations of cells within the human ovary that do have the potential to form what appear to be oocytes. So these can be isolated from the adult human ovary, and whilst this is still a very controversial area, the results that we're obtaining are encouraging, and the presentation highlights the need for further characterization of the cell types that exist within the adult human ovary. And then the fourth presentation was by Professor Kawamura from Japan, and he highlighted the process of in vitro activation, so IVA, to activate the growth of primordial follicles in patients where they have very few primordial follicles. And he spoke about manipulating the PI3K pathway, particularly using pharmacological agents to activate the PI3K pathway, and then also to inhibit the P10 part of the pathway, that are all involved in regulating the activation of primordial follicles. So by doing this prior to transplantation, they're able to obtain greater activation of primordial follicles, and they have been able to obtain pregnancies using these techniques. Professor Kawamura went on to discuss the non-pharmacological activation of primordial follicles, so using mechanical methods to induce the Hippo signaling pathway. And these look very promising and being able to achieve similar results to the pharmacological factors. And finally, to end this session, we had Dr. Meena Chowdhury of University of Newcastle, and she gave an update on the status of pronuclear transfer in human embryos to prevent the transmission of mitochondrial DNA disease. And Newcastle was the first centre in the UK to obtain a licence from the Human Fertilisation and Embryology Authority in the UK in order to develop these techniques. And Dr. Chowdhury gave a very comprehensive overview of the technical challenges involved in this technique, as well as covering the ethical and legal issues before moving forward in this incredibly important area.
Evelyn, thank you very much for such a very good summary. And we will move forward to the last session of this meeting.